morning students. Already we have discussed about plant tissue culture. The plant tissue culture is a alternate technology which we are using to make a large number of desired plants in less time and less space. Understood students? In that we have a very small topic is remaining. Today we are going to discuss about the remaining part of the plant tissue culture and one more important method what scientists are following to make different plants what they are producing through the laboratory conditions, that is called somatic hybridization. So today we are discussing about the remaining part of the plant tissue culture what we have, that is about meristem culture and somatic hybridization. What are the methods and techniques are very much useful for the growing different desirable plants, okay, for our convenience sake, for getting more yielding and high productivity, food productivity what generally we are producing. Understood students? So let us know something about that small remaining part of the tissue culture, that is okay, meristem culture. You please put the subheading. Today topic is meristem culture. What it is? Meristem culture. You please put the subheading because it is the subtopic of tissue culture. So meristem culture. Today's topic is meristem culture. Okay. What is the importance of meristem culture and how we are going to do that meristem culture? It is also same as it is what the methods and processes we followed in the tissue culture. Here we are observing same. Okay, it is also the method as it is we follow to do like tissue culture. What is the method or processes we follow in the tissue culture as it is we are following that meristem culture also. So meristem culture means before going to discuss about the meristem culture, what is meristem? I think you know in your previous class you learned very well about that okay, term meristem. Meristem means it is the capability or ability of plant cells where they are going to divide rapidly, where the number of plant cells are going to be divided rapidly and producing a large number of cells which are basically need to make tissues and organs. Okay, dear students, in the plants only we just observing the cellular organization is maximum up to organ level only. We are not observing any organ system like human beings and some other well developed animals. Okay, that type of development and cellular organization we are not seen in the plants. The maximum development of the or maximum cellular organization we seen up to organ level in plants. Understood students? So see here that organ level is only built up and developed and produced and formed only because of the cells which they are producing as basic units of construction of the body building of plants through ability and capability is called meristem. Meristem is the property of ability or capability of plant cells. Plant cells, the plant cells which they are going to divide rapidly. The plant cells which they are going to divide rapidly. Now you got the point students? Where the plant cells are going to be divided rapidly. Okay, wherever you may check, for example, plant cell 1. The plant cell 1 which has ability of meristem, then it is going to divide into two. Again these two individually, independently, they divide again and again with the particular ability and capability of meristem. Then these 2 cells into 4, 4 cells into 8, 8 cells into 16, like that a large number of cells, okay, large number of cells, a group of cells they developed. Understood students? Large number of cells are developing only through the, note down students, only through a property of meristem. What it is? Meristem. You please note down, students. Yes, based on this property meristem only, a number of whatever the cell we are observing, whoever they may be, whatever the living form it may be, okay, whether it is unicellular or multicellular, just only they started their life from the single cell. If it is unicellular organisms, their body or the living structure completely with a single cell, of course, because that composition of cells are not seen, they are completely divided into individual organisms in unicellular organization. But in the multicellular organization, what we are observing, cell to cell they make grouping of cells which they are calling as tissue. Understood, students? And grouping of tissues collectively called as organs. Different organs we generally seen in the body of young plants and animals. Understood, students? So whatever the organ development we seen or formation we seen in the body of an individual, either plant or animal, just only they started with a single cell only. Whatever the life of organisms, whether they are multicellular or unicellular. Here we are discussing about plants. Here we are discussing about plants. Plants are the multicellular organisms, you know. Understood? Okay, especially well developed plants which they are going to show organ level, whatever it may be, organ level. What is that? Organ level. So in the plants we generally seen maximum level of organization of their body is organ level only. Organ level. To make the number of organs, to develop a number of organs requires a number of cells and number of tissues. Tissues and organs are nothing but completely they only constructed by cells, basic units like cells only. That cell number is needed to increases by following the method of cell division. The rapid cell division is only taking place by the property of plants and plant cells, which we are calling meristem. Understood? The cells which possess an ability or capability like meristem is called meristematic cells. What the cells they are? Those are called meristematic cells. Those are the cells we are calling what, Nana? Meristematic cells. These meristematic cells are found rich in amount. Note down, students. Rich in amount of meristematic cells. Meristematic cells we seen in shoot tip, root tip and axillary buds. And in the regions of axillary buds. So these are the selected parts in which we are getting meristematic cells. Okay, for our process to perform the process of meristem culture, that meristem culture which we are using used as tissue culture, you know the result of tissue culture always producing what it is, producing a large number of clones. Clones are nothing but, you know, young generations or young plants what we are producing. Now we got the point. We are producing young plants through the method of tissue culture. Here the tissue culture what already we discussed. Yes, now here we follow another method of tissue culture like meristem culture. What is the speciality of this particular process like meristem culture once we focus? Understood? So it is also a type of tissue culture. Then why we are discussing separately? Okay, we have another importance to do this process of meristem culture, that is we are producing clones. Meristem culture. Those producing clones are free of pathogens. Means free of pathogens means this plant is not having that resistance, of course, but we are producing plants free of pathogens from diseased plants. From where we are getting these plants? Diseased plants. From diseased plants also we are getting non-infectious, free of diseases, young plants we are producing. Now got the point? Whatever the young plants we are calling clones, these clones are pathogen free. What it is, students? Pathogen free or free of pathogen or disease free. You please remember, students, what it is? Disease free. Whatever the young plants we are producing or young plants which we are calling what? Clones. Clones, when we use the term clone, when young generations which are genetically identical to the parental plant or parental organism, then those are called clones. Now you got the point? Then that particular offsprings or generations we are calling only as clones. Understood? Especially here these clones we are using what? Somatic parts. All these are the parts like, for example, shoot tip. What it is? Shoot tip or root tip or and axillary buds. All these are the somatic parts or body parts. Here we are using body parts only na, somatic parts only na. For the culture, what is the culture? It is a tissue culture. As one of these process of tissue culture like meristem culture, because we have taken the tissue which we are calling meristematic tissue or meristematic cells, we just collect them and culture them. That's why this process we are calling what? Meristem culture. For what purpose we are growing these meristematic cells? Just for the process of just for the production of young generations as clones which are pathogen free, which are disease free. Now got the point? Means what it is? Suppose to take a plant we have understood in our crop field or farm field, we have a variety, a good variety of plant we have understood, but that plant is suffering from disease or it is completely infected by disease, understood, but it has ability to give more and more yielding and very good quality nutritive food it is giving, but it is infected and it is diseased. Then how we are going to produce the plants from the same plant without disease, or to stop the disease? How we are going to produce young copies? Just we produce young copies with free of diseases, with free of pathogens, with a high yielding quality what we are expecting only from the parental plant, that is only we are obtaining from the diseased plant. So though they are diseased plants, we are producing young plants, young generations is as clones which are pathogen free or disease free. Now got the point? Pathogen free and disease free. That type of young plants we are producing, that particular large number of population what it is producing are called clones. Understood? But here we are using all these are the parts what we are using for the meristem culture are somatic parts. Means meristematic cells are nothing but what it is, meristematic cells are the somatic parts. These somatic parts we are using for culture na. Culture. These somatic parts are meristematic parts or meristematic tissues or meristematic cells we are using as for our culture for the production of clones without disease. Understood? The disease. From which plant we are getting these meristematic tissues? We are getting these meristematic tissues or cells from the diseased plant. Why should we get for production of young generations with disease free and pathogen free? Because we have a required character like high yielding where it is present in the plant which is a diseased plant. Though the parental plant is diseased plant, but the young generations are not having or carrying the disease or disease causing organisms like pathogens. The pathogens and diseases are not carry forward into the generations what we are producing by following the method of tissue culture like meristem culture. Whenever we are going to collect the meristem from the diseased plant, that meristem does not give the opportunity for the survival of pathogens, especially like viruses. Understood? Survival of the pathogens, especially like viruses, may not grow because in the meristematic cells where they are present in the plant body like root tip and shoot tip, have a high concentration of auxins and have a rapid cell division or high rate of cell division we are observing in the meristematic region. So due to that those two regions, like what the regions they are, high concentration of auxins and high rate of cell division or rapid cell division what we seen in the meristematic cells where they are present in the plant body like root tip and shoot tip and axillary bud. So in those from that only we are getting these particular different kinds of meristematic cells we gathered, we collected, though they are collected from the diseased plant that from which we are producing young generations or clones are not, may not carry the disease disease, okay, or pathogens may not enter into the younger generations what we are producing from the meristematic cell what we have collected from the diseased plant. So from the diseased plant also we are producing young copies as the clones which has ability to give more yielding or with desirable character. On which character we are doing, for which character we are doing this method of plant tissue culture like meristem culture for the production of young copies as plants called as clones which are pathogen free or disease free. Now got the point? Understood, students? So these are the different kinds of different clones we are producing, different clones with free of pathogen, free of disease we are producing through the method of tissue culture called meristem culture. Okay, the meristem culture, why should we go for producing clones without disease or without a pathogen for the production of desirable plants? Understood? We have desirable plants in our surroundings, of course, but desirable character like high yielding they are giving, right? Understand? But they are completely diseased, completely infected by diseases. If they may completely what we call destroyed by the diseases and disease causing organisms, what happens ma? So it is not possibility to give more yielding, though they have their own character, their own parental character like high yielding, but that metabolic functions are not in normal conditions due to deviations created by that particular disease causing organisms. That's why we want to producing young copies with a desirable character like high yielding without diseases, without pathogens. That type of plants and plant production as clones we are producing from meristem culture only. That meristem we are taken out from the different regions of the plants like shoot tip, root tip and axillary buds, which they contains more amount of meristematic cells which are not give the opportunity for the survival of viral pathogens. Understood? Then though the entire plant body or diseased plant body is suffering from viral particles or disease causing organisms, but in the regions of meristematic regions or meristematic cells may not give the opportunity for the deposition or accumulation and survival of pathogens. That's why that particular disease free cells and regions we have collected from the diseased plant like regions of meristematic regions. Meristematic regions, especially in the plant body, I think supposed to take, it is a plant body, understood? It is a stem part which differentiated into nodal regions, right? Understand? So this is the root part, this is the shoot part, understand? Here two regions we are observing as terminal regions, understood? In between these two parts like part 1 and part 2, supposed to take. In the part 1 like shoot tip, the terminal point which we are calling what ma? Shoot tip. What it is? Shoot tip. In the same way here part 2, it is a root na. That root is also having a terminal point. The terminal point which we are calling what ma? Root tip. What it is? Root tip. So in these two regions, higher amounts of meristematic cells we seen, which are free of pathogens, which are free of diseases. Now got the point? And here all these are the regions which they differentiated that particular apical part of the or whatever the outer aerial part what actually we see in the plant body like shoot differentiated into nodes or junctions what we see in the shoot stem. These particular nodal regions some other extra growing parts are going to be as buds will be developed. These buds which they are developing on the node from the nodal portions on the stem is called axillary buds. What they are, students? Axillary buds. These axillary buds are also very actively growing parts. Very actively growing parts means they contains meristematic cells. Wherever the growth we seen in the plant body, that growth is only influenced by meristematic cells. See here, the tip of the shoot and the tip of the root are very much young growing parts of the plants and they are rapidly growing parts along with axillary buds. Means what? Shoot tip, root tip and axillary buds contains more amount of or more number of actively dividing cells called meristematic cells. That cells we are collecting from the diseased plant and producing non-diseased young generations or disease free clones we are producing or pathogen free clones will be developed. These clones are developed from or by using somatic parts. Whatever all these are the parts are come under what it is? Somatic parts or body parts, you know. What these are? These are called somatic parts, isn't right, or somatic cells, somatic structures. So, due to that, by using that somatic parts, we are producing young clones. That's why these clones are also called as somatic clones. Somatic clones. Whatever the younger generations we are producing, whatever the younger generations we are producing from the method of tissue culture, all those are collectively called as somatic clones. You please remember, students, and note down somatic clones. Understood? So, these somatic clones we are producing by using meristem culture. What it is? Meristem culture, right? So, this is very important for the production of young generations, what we are producing by following method of meristem culture. Here also same, what process we following in the plant tissue culture. What process we following in the plant tissue culture, here also we follow same. Okay. During plant tissue culture, what are the methods we have follow? We follow first select desirable plant. Okay, what is the first activity we seen here? Desirable plant. Desirable plant. Here also same methods we follow. Understood? Desirable plant. The second one is from the desirable plant we are going to get collect explant. What it is? Explant. Here in the normal tissue culture, what we made maximum, that is any part of the plant which has ability to divide. Any part of the plant we have collected for tissue culture. Understood? Which has ability to divide, or if they may have it, if they may not have ability to divide, they are going to give some artificial facilities to change their ability, to change their behavior. Then all the cells are going to be divided, multiply to give whole plant. But here what we seen here, explant only, specially we are collecting that explant like meristem. What is the explant we have chosen here? Meristem we have chosen here. Meristematic cells or meristem we have chosen as explant. So, that meristem is only we are collecting from diseased plant. From where we are collecting? This meristem we are collecting from diseased plant. Understood? Why should we get diseased meristem from diseased plant? The diseased plants which has a desirable character like high yielding. Not from all the diseased plants what unnecessarily we are collecting. All the diseased plants are not useful for us. Understood? Commercially or economically cultivated crops in which we seen a desirable character like high yielding. Understood? And growing very well to give more yielding, though they have ability to grow well, if they may easily suffering from diseases, that particular diseases plants for getting high yielding character in the younger generations, to produce younger generations as clones with free pathogen or disease free plants what we want to produce, then we go for the method that is meristem culture. In the meristem culture also we have to choose a particular structure of the plant like meristem only. Okay, here meristematic cells where they are present, we are obtaining from the plant body like shoot tip, axillary buds, especially to shoot tip and axillary buds we have chosen to collect from the plant, that is that particular collection of the materials is as explant. What it is? Explant. This explant we are now placing this explant over the nutrient medium. We are placing the explant on or over a culture medium. Now got the point, students? So here whatever the explant we have collected from the desirable plant, simply we collect and place them on the nutrient medium. Understood? Before that you know very well what are the activities we have done in the process of tissue culture. As it is we follow here explant what we have chosen, we have to follow the process of sterilization. What it is? Sterilization. Whatever the activities, whatever the conditions you are observing in the tissue culture, as it is you have follow. Here explant like either shoot tip or root tip. Root tip is very rare we are collecting, of course, but axillary buds also very familiar. just we are using as explants. Now got the point? These explants we have to do sterilization. Sterilization means what? The outer surface of the explants we have to wash properly with antimicrobial chemicals or or whatever the chemicals they are going to clean or wash the pathogens which they are present on the surface of the explant. That's why that sterilization what we have done on the outer surface of the explant, that's why that sterilization we are calling what? Surface sterilization. Already we discussed surface sterilization. What the sterilization method we follow, that is surface sterilization. It also called as external sterilization. It also called as external sterilization. External sterilization we follow. So external sterilization we follow. So whatever the surface sterilization or external sterilization what we have done, for example, whatever the meristematic cell if you may take out from the diseased plant, suppose to take it is a plant cell or group of cells from the meristematic tissue or group of cells what we have collected from the meristematic region. Okay, suppose to take it is a plant cell. We just to understand better, just we focus on one plant cell. It is a plant cell. Okay, one plant cell. If we are going to wash it properly on the surface of this plant cell with antimicrobial chemicals, just we wash and clean them. Okay, the surface of the plant cells and tissues like meristematic cells, that is we are calling surface sterilization or external sterilization. After completion of the external sterilization or surface sterilization, then only that explant we placed them on the nutrient media. Before that this nutrient media or culture medium is also should be sterilized. Should be sterilized. Already you know how we are going to do the sterilization process. Sterilization process on non-living materials. So here these nutritive materials and different containers, different instruments and vessels we are using for the tissue culture, all those are going to be complete sterilization. Should be give complete sterilization. Complete sterilization means just we wash all the microorganisms and stop the growth of the microbial colonies. Just simply we stop them by changing the atmospheric conditions, by sending this nutrient media for getting proper sterilization or complete sterilization by sending it to the autoclave method. It is a very good method, autoclave method. So in autoclave method, what actually we are maintaining the climatic conditions like they are maintaining this autoclave of all these nutrient material just we done. Here only the culture medium, we are discussing about culture medium. Yet we placed it in the autoclave at the 120 degrees Celsius and 15 pounds of pressure. 15 pounds of pressure. 15 pounds of pressure and for nearly 15 minutes time we have to place them. Understood students? We have to place them in the 15 minutes time at 120 degrees Celsius. Then hot air will be produced. From the autoclave they are giving hot or steam, hot vapors or hot steam is going to be clean the culture medium. Then that culture medium is suitable for giving supplement of nutrients to the explant what we are ready to placing. Before the placing of explant on culture medium, that culture medium should be get proper sterilization. That is we are calling complete sterilization, and also that explant is also came from the what it is external sterilization or surface sterilization. After that only these two are the sterilized materials like explant and nutrient media in which containers we have taken. All these should be get a proper sterilization. After that only these two are going to be after that only the placing of explant we have done. after the placing of explant, then that explant is going to absorb nutrients. What it is, students? Absorbs nutrients from nutrient media. Already we discussed about nutrient media, what it contains. What it contains, students? Nutrient media contains, okay, different kinds of organic sources like sucrose. It contains sucrose as chief sources of food. Understood? For young growing meristem into baby plant. Sucrose and different vitamins we are adding here. Understand, students? And amino acids are there. Okay, it contains amino acids. Note down, students, amino acids and okay, inorganic salts also we have to add. Inorganic salts like minerals. Inorganic salts we have added it into this. Understand? And different kinds of essential nutrients they are going to be added to the particular nutrient media, which is going to maintain a pH, which maintain pH is 5.7. Whether this nutrient media, it may be liquid or solid. It may be liquid or solid. Nutrient media we are using here, liquid or solid. Now got the point? On that nutrient media we are placing explant. After that we have to give time nearly 3 to 4 weeks, of course. After the inoculation of explant, we have to, inoculation means placing of sterilized explants over a sterilized nutrient media. Now got the point? All these are only done in aseptic conditions. Everything we done in the aseptic condition. Aseptic conditions means pathogen free. Aseptic conditions means pathogen free conditions or laboratory conditions. Now got the point, students? Laboratory conditions, you please remember. So aseptic conditions are laboratory conditions. Whatever the activity we have done, all those are the activities done only in the aseptic conditions are laboratory conditions. Now got the point, students? So like that, whatever the processes we are following, all those are the processes what already we have discussed in the tissue culture, as it is we are following. But the difference is the explant is not collected from the plant, from any part of the plant. We are going to collect the explant in the meristem culture. We only collect the explant from the specific regions of the plants like meristematic regions. Now got the point? So meristematic regions are generally they are present in the plant body like root tip and shoot tip and axillary bud. Mostly we are using that axillary buds as meristematic cells or meristematic parts which we are using as explants or shoot tip. Shoot tip or axillary buds are the more superficial okay, structures we are using as explant which we are growing on the nutrient media for producing young generations. Okay, finally what after the absorption of nutrients simply they are growing into callus. Already I said callus means callus is the undifferentiated proliferating mass of cells what they are continuously producing from the rapid cell division which done in the cell what we placed as explant on the nutrient media. Whatever the explant we have taken place, whatever the explant we placed, that explant by absorbing nutrients in the time incubation period of time or incubating time 3 to 4 weeks, it absorbs nutrients, it absorbs different kinds of inorganic salts and organic sources and also phytohormones. Already I said na, PGRs, plant growth regulators. Plant growth regulators especially we are taken in the tissue culture like auxins and cytokinins. You please note this point. Auxins and cytokinins. Okay, the artificial auxins like 2,4-D we are using. Okay, artificial cytokinins like BAP we are using. What it is? Benzyl aminopurine. Now got the point, students? So those two are the synthetic phytohormones we are adding into the nutrient media, from which media the plant is going to be, our explant is going to be absorbs nutrients for its growth and development in the period of time 3 to 4 weeks what we have given to the explant, after the inoculation of explant on the over the nutrient media. In that particular time, it is going to producing a large number of undifferentiated, large number of unorganized, organization is not here, there is no organs of root, stem, leaf, but crowd of cells will be developed, a number colonies of cells will be developed, that is we are calling what callus. From the callus we are expecting to produce according to the concentration of the phytohormones, according to the changing of nutrient media. Whenever the person, a boy supposed to take, okay, 2 years boy, their nourishment is completely differentiate with the, okay, young boy. So in between 15 years old or 25 years old, so where the age difference we are observing, the requirement of nutrients is also different. In the same way, here only one cell we placed as explant on the nutrient media. There in the initial at the first time of the nutrient media on which we placed the explant first initially, there the nutrient media contains nutrients are completely different with the, okay, nutrient media what we provided to the young plant, what it is comes after the callus. Understood, students? So that's way we have to change the nutrient media from stage to stage. Stage to stage means explant to callus, callus to organogenesis, what it is taking place to produce organs like root, stem, small leaves, young leaves, what they come out. So like that a number of stages we are observing in the plant and its growth, dear students, in the lab. So according to the stages of growth in the plant body, we have to alter or change the nutrient media and their concentration, which is the concentration, which is the nutrient media is the suitable media for the growth of that particular stage of the plant body. So that's why we have to alter the nutrient media and producing finally a large number of plants. A large number of young plants, which we are calling as, which generally called as, okay, Soma clones. What they are, students? Soma clones. These Soma clones what we developed from Meristem culture. What we developed from Meristem culture is young plants is pathogen free or disease free. Pathogen free or disease free. Now got the point? This young population what we are producing from the Meristem culture as one of the methods of tissue culture gives a large number of colonies, a large number of population or young generations, which we are calling Soma clones, which are collected from the diseased plant. Though we are collecting meristematic cells as explant from diseased plant, but we are not producing clones with disease. Now got the point? We produce clones without disease or without disease causing organisms with desirable character, which it is present as inherited character in the parental plant. So we can easily to produce a large number of clones without a disease or disease free or pathogen free. Just we go for the method of tissue culture, which we are calling Meristem culture, which is a very much beneficiary method of tissue culture through which we are producing a large number of okay, Soma clones without disease or disease free or pathogen free plants we are producing. If you want to producing a young population as the Soma clones without disease from the diseased plant or from the diseased parental plant, if you want to get more population without disease or disease free or pathogen free plants, then we go for a method of tissue culture, Meristem culture, through which we are producing okay, all those are the young plants with disease free or pathogen free, okay, properties. Then it is very much useful for producing a large number of plants, especially in the commercially valuable plants like potato, sugarcane. Note down, students: potato, sugarcane, and in the banana, the meristematic cells of sugarcane, the meristematic cells of potato and banana, we collect it and follow the processes as it is to produce young copies without disease. So for this purpose, we go for method of tissue culture like Meristem culture, which is very much useful for producing young generations without disease, or the young generations which are not carrying pathogens for express the disease. Now got the point, students? So it is very much beneficial method through which we are producing so many plants in the less time and less area without diseases. What we are producing, especially in the commercial, economically important cultivated crops are food crops like banana, sugarcane and potato. Understood? Next we are going to discuss about another method which is very much familiar to produce different kinds of plants as with desirable characters, which we are calling hybrids. Okay, here to produce these hybrids we are not following any sexual mode, here only they are following somatic parts or body parts or somatic cells or vegetative cells only. By using that vegetative cells they are producing different kinds of varieties, that is we are calling hybrids. These vegetative characters also called as somatic characters, those hybrids what newly we are producing. Okay, by using these somatic parts, that's why those hybrids are called somatic hybrids. That process we are calling somatic hybridization. So now we are going to discuss about somatic hybridization or protoplast fusion. What is the next method? Protoplast fusion. Fusion means combining. We have to union two different cells and their information, genetic information and cytoplasmic information we have to fuse to form a single cell, that is we are calling what? Hybrid cell. What it is? Hybrid cell with two different characters. Genetically different characters are there in the nucleus or nuclei collected from two different cells. We have to fuse them and make a single cell which has desirable characters. That cell is called hybrid. These two are somatic cells. That's why that hybrids are also called as somatic hybrids, while the process is called somatic hybridization. Have a look into the somatic hybridization or protoplast fusion, how we are going to do and what is the benefits we are obtaining from the somatic hybridization or protoplast fusion. Okay, students? Now, we discuss about another method which is also very much important for producing different plants with hybrid characters or desirable characters. That is the method we are calling what, ma? You please put the heading, that is protoplast fusion. Protoplast fusion. Fusion means combining or union, mix. That is the meaning of fusion. Protoplast fusion are also known as somatic hybridization. Somatic hybridization. Somatic hybridization. So this is our topic. What we are going to discuss? This is the topic. What it is, students? Okay. Protoplast fusion or somatic hybridization. See, what is the process through which what kinds of information we gather to do, that particular experiments what we have done in the laboratory conditions. See here, protoplast fusion or somatic hybridization is very important process for producing young copies of, we are going to producing young copies of plants. Young copies of plants with desirable characters. With desired characters. Desired characters. See here, to do this process, to get the desired characters in which plants we are producing, if you want to producing them, we just go for preparation of protoplast or protoplast preparation. What should we do? We have to do protoplast fusion, protoplast preparation. Protoplast preparation means what is protoplast? Protoplast is nothing but it is the process completely related to the plants. Understood? So that's why we have to take this particular to do this process. We have to take plant cells. What are the cells we have to take? We have to take plant cells to produce young copies of plants with desirable characters as a hybrid. Okay, so this is the plant cells. Whatever the plant cell you may have taken, the plant cells have the outer wall or outer layer which we are calling as what it is? Cell wall. What is the outermost layer of the plant cells? Tell me. I think in your early classes you have learnt very well about the plant cells. You have learnt very well about the plant cells. Okay, this is the outermost particular region. This is the particular structure we are calling what layer, which is called cell wall. Understand? Okay, it is cell wall which is completely covers the inner layer or inner membrane, which we are calling what it is plasma membrane. It is a plasma membrane. Plasma membrane. You know the remaining different cell conscients they are present. Different cell conscients and cytoplasm which we are observing here. Everything is here. Understood? Suppose in the plant cell the maximum area occupied by a cell organelle called vacuole. You know, this is a small spherical body like it is called nucleus, which present at that slightly away from the center. The central maximum portion is occupied by vacuole in the plant cells, but slightly away from the center we have another a small spherical structure which we are calling nucleus. These are the structures what generally we see in the plant cells. This is the normal plant cell, but this is the plant cell we have taken as our material. But what kind of material is needed here? Protoplast. So this plant cell, when we remove cell wall, okay, or removal of cell wall, where is the cell wall? Cell wall is the outermost layer which we see in the plant cells. This outermost layer like cell wall, now we want to remove. The remaining, all these are the regions and conscients as it is present. That particular naked plant cell means without jacketed wall or without outer layer like cell wall. That particular conscients of the plants or plant cell is called what it is? A naked plant cell is called protoplast. What it is? Protoplast. So after the removal of cell wall, we are going to make another small conscient like our second membrane, which we are calling already here we discussed plasma membrane. Because we remove cell wall, just we have now plasma membrane along with the another conscients what as it is they are present in the plant cell along with the vacuole and nucleus and other all cell organelles as it is they are present in the cytoplasm. So now this is the particular structure we are calling what protoplast. This is the structure what we are calling it is protoplast. Two, what is the difference between the A and B? A is a normal plant cell. But here what happens here? That normal plant cell lost cell wall. The outermost layer like cell wall is not present here. That cell wall is not present here. That cell wall is completely removed, destroyed. The cell wall is completely removed. This is the cell wall. Now cell wall is completely lysed or destroyed. After the removal of cell wall, whatever the cell wall is going to be removed, here cell wall remove. After the cell wall separation, we have a inner membrane along with the cytoplasmic conscients which they are present is called protoplast. Okay, this is the process we have to done. To done this job, we require some enzymes. You please note down, students. Requires some enzymes like cellulase and pectinases. Cellulase and pectinases. You know very well, yes students, the composition of cellulose is more in the cell wall. You know very well. Understood? Some other pigment, what we call polysaccharide materials also we are observing like what it is, pectins. Pectins are also be seen. So to remove the cellulosic materials and pectin materials, we just using that specific hydrolytic enzymes like cellulases and pectinases. Now got the point? Hydrolytic enzymes like cellulase first break down the cellulose or cellulosic substances. Okay, pectinases is the enzymes more suitable for the lysis of pectins. Understood? Which they are present in the cell wall. Due to by using these two enzymes like cellulase and pectinase, cellulase and pectinases, it is the cell wall is easy to remove. The cell wall is easy to remove and change the normal plant cell into and we change the normal plant cell into protoplast. Now we change the normal plant cell into protoplast which doesn't have cell wall. Now we got the point. So this is the process what we have done in the laboratory conditions. It is called protoplast preparation. You please note down, students. This is the process we are calling what protoplast preparation. After the protoplast preparation, what should be done? What we have to do? Okay, we have to do the process of protoplast fusion or somatic hybridization. This is the process we done to produce different varieties of or different species of plants. Understood? For desirable characters. Now got the point? So whatever the process like protoplast fusion, we require protoplasts. These protoplasts we collected from two different we made this protoplast fusion in between two different varieties. Of plant cells or two different species or two different genera. Genera, just we have to collect the from in between two different varieties or two different species or two different genera. Variety 1 versus variety 2. So two different varieties. This is we are calling what intervarietal protoplast fusion. Intervarietal protoplast fusion. In the same way, two different species A and species B. Two different species also we are using for getting protoplasts. Understood? So it is also called as interspecific protoplast fusion. Understood? Or interspecific somatic hybridization. In the same way, genera. Okay, intergeneric protoplast fusion we are observing. Whatever the different kinds of species or different kinds of varieties, different kinds of genera we are using to we follow to do somatic hybridization or protoplast fusion. Means what here? Two varieties means two different variety of plants we are using. From that plants only we are collecting individual plant cells only we have to collect individual plant cells. From variety 1 we have collected some plant cells. Okay, which have desirable traits. In the same way, variety 2 also, from variety 2 also we have taken some plant cells, some cells which they have desirable characters. Now those are the plant cells, we collect them and used as chief materials or main materials for changing those collected plant cells into protoplasts by following the method of protoplast preparation by using enzymes like cellulase and pectinase, where the plant cells we have taken into our lab, then we are going to remove their cell walls and change them into protoplasts. Now those protoplasts we are trying to artificially in the laboratory conditions and aseptic conditions and in the sterilized conditions we have to fuse them. We have to fuse them. Fuse them means we have to combine those two plant cells or protoplasts which we have taken from two different variety or which we taken from two different species or which we have taken from two different genera. You got the point? Whatever the different varieties of plants we have selected as parental plants from which we have taken those plant cells, then we have to prepare them as protoplasts. Those protoplasts only we are using for the process of hybridization, that is somatic hybridization. What it is? Somatic hybridization. Now got the point? So this is the particular activity. If we want to do the process of protoplast fusion or somatic hybridization, how we are going to utilize these protoplasts, let us see. Okay. To do now, we have a look into the protoplast fusion. Protoplast fusion. Okay. Protoplast fusion is not easy. To do the protoplast fusion requires two different activities or two different techniques we follow. Whatever the plant cells we have taken, the plant cells we change them into protoplasts. That protoplasts are not easy to mix one another. Okay, they want to mix. If we want to mix them, we should follow to use some chemicals or some physical factors. Understood? Those are called, okay, to perform the process of protoplast fusion, we follow what it is, okay, electro fusion. Electro fusion, and second method what we follow to do the protoplast fusion, that is chemo fusion. Students, are you understanding? Here these two are the techniques, these two are the factors we are using to do the protoplast fusion. First one is electro fusion. Electro fusion means what it is? Just we are using some electrical factors or sources. What it is? High frequency alternating electric field with short current pulses. Okay. Electrofusion means here some electric charges we are using. Some electric charges and electrically some artificial climatic conditions provided to the two protoplasts, whatever it may be, we have taken from two different varieties or we have taken from plants of two different species or of plants of two different genera. Whatever the protoplasts we made, those protoplasts we trying to fuse or combine or union or mix them. Understood, students? So to fuse these two different genetically different and diversified protoplasts, just we are using some artificial conditions we provide. In that two are there. What it is? The first one is electrofusion and second one is chemo fusion. Electrofusion means we are using here electric field by high frequency alternating electric field with short current impulses, or whatever the movements are going to be generated for the fusion of two different protoplasts. That is, if you are using electric charge, then that is called electro fusion. Then coming to the point, or otherwise, in some cases they are following electro fusion to fuse the protoplasts, two different protoplasts, or otherwise, in some cases we follow another method, that is chemo fusion. Chemo means chemicals. Chemo means what it is? Chemicals. Some chemicals we are using to change the atmosphere and our environment or vicinity of the protoplasts. The protoplasts in which container we have taken, into the container we have to change or provide artificial climatic conditions, through which conditions those two genetically diverse protoplasts are going to be mixed, are going to be fused, are going to be united. Now you got the point, getting union. So that is the chemicals we are using, especially like PEG. In between these two processes, any one of the processes we follow to do protoplast fusion. Understood? Either we go through the electro fusion or either we go through the chemo fusion. So chemo fusion, in the chemo fusion we are using chemicals like PEG or sodium nitrate. PEG or sodium nitrate are the chemicals what we are using to fuse the protoplasts. Now got the point? Here PEG full form is what it is? Polyethylene glycol. Note down students, polyethylene glycol. Polyethylene glycol short form is PEG, whatever it may be, either PEG or sodium nitrate we are using to fuse two different protoplasts. Understood? So by using that particular factors, by providing that particular factors like either electrically or chemically, means electro fusion or chemo fusion, after finishing that fusion, whatever the fusion we have done through the chemicals, by using chemicals or electrical field, just we made complete protoplast fusion. That protoplast fusion is generally we are calling that a new cell what it formed after the fusion of two protoplasts are called hybrid. What it is? Hybrid. Understood? In this particular hybrid we are observing what it is. The cytoplasm is going to be fused. Now they are ready to the nucleus or nuclei of two different protoplasts are ready to fused. Understood? So the first one is cytoplasmic fusion we are observing, followed by nuclear fusion. Now nuclear fusion. Cytoplasmic fusion and nuclear fusion completely gives a single cell which we are calling hybrid cell. What it is? A hybrid cell. Here in the hybrid cell we are using what? Somatic cells we are using. That cell is the somatic cell. That's why it is that hybrid cell which we are calling, also called as a somatic hybrid. What it is? A somatic hybrid or somatic hybrid cell. While that process is called somatic hybridization. That process is called somatic hybridization. Okay, through the somatic hybridization, we are going to produce what it is? Hybrids. Okay, those are come from the somatic parts, somatic cells. That's why those hybrids are called somatic hybrids. What it is, students? Somatic hybrids. Understand? Or in the somatic hybrid, cytoplasm is also involved, fused. That's why those are also called as cytoplasmic hybrids. Cytoplasmic hybrids. Now got the point? So cytoplasmic hybrids. These cytoplasmic hybrids are also called as hybrids. These cytoplasmic hybrids are also called as hybrids. Now got the point? So finally, by using this electro fusion or chemo fusion, just we are going to fuse or combine two genetically diverse protoplasts to form hybrids. hybrids called somatic hybrids. Somatic hybrids. While the process is known as, note down students, while the process is known as somatic hybridization. Somatic hybridization. Note down. Somatic hybridization. Now got the point? So finally we are producing what? Somatic hybrids. That somatic hybrids we produce through the process is called somatic hybridization. With the desirable characters we are producing. So these somatic hybrids may be okay in the after the process of chemo fusion or electro fusion, we are observing that somatic hybrids may be syncaryon or may be heterokaryon. Syncaryon means okay, immediately after the cytoplasmic fusion, those two nuclei are going to be fused to make a single nucleus. Understood? Or in some cases that particular single cell is single, but after the cytoplasmic fusion these two nuclei are getting fused by taking time. In that case we are observing two nuclei are present in one cell. That condition we are calling what? Two unfused nuclei are present. Two unfused nuclei we are observing. That condition we are calling heterokaryon or heterokaryon hybrid, what we are observing. Understood? Later that heterokaryon means two unfused nuclei are also getting okay fused, or otherwise okay simply the cytoplasmic fusion and nuclear fusion, after that they are behaves like individual hybrids. Understood? Or in the syncaryon only one fused nucleus is present. Understood? So somatic hybrids may be syncaryon or heterokaryon, which they are going to produce young copies with desirable characters as hybrids. Genetically diverse plants they are producing. Here there in the plant tissue culture already we have discussed about plant tissue culture, okay, and meristem culture are very much superficial for producing clones which are genetically identical to the parental plant. But in the somatic hybridization or protoplast fusion, whatever the young generations they are producing are not genetically identical. That's why those are not called as clones. Those are called as hybrids. From the somatic parts only they are producing, that's why those are also called as somatic hybrids. They are producing through the process is known as somatic hybridization or protoplast fusion. Understood students? Protoplast fusion, through which we are getting desirable somatic hybrids which are very much beneficial for producing young generations with desirable characters. Now got the point? So many economically important cultivated crops are producing through the method of somatic hybridization or protoplast fusion. Understood? Right students? So some exemplary somatic hybrids what they are producing by scientists, have a look. So through this diagrammatic representation we understand the protoplast fusion, how it is going to do. Understood students? See here, for example, we are going to take that plant cells from two different varieties, or plant cells from two different species, or plant cells from two different genera. These three are the opportunities of plant cells what we have taken to produce desirable hybrids. Understood? See here. So by this diagrammatic representation we can easy to understand. For example, now we are using two different protoplasts. Two different protoplasts. Two different protoplasts either from two different varieties or either from two different species or either from two different genera. Understood? Two different genera. We may take from okay genera 1, genera 2. Understood? Genera 1 or genera 2. From genera 2. So whatever it may be, two different kinds of plants we have chosen to take plant cells, and after that plant cells, you know, already we discussed these plant cells, normal plant cells, we prepared them into protoplasts, you know, we prepared them into protoplasts. Protoplasts. How we are going to prepare the plant cells into protoplasts? By using lytic enzymes or lyse the enzymes what they done to remove the cell wall. That is what it is, pectinases and cellulases. Pectinases and cellulases we are using. After that we are producing protoplasts. These protoplasts we collected from two different parents, like for example parent A and versus a parent B. Means two different parents we have selected. These parents are selected parents. Selected parents means desirable characters in which plants they are present with higher proportions. Understood? More required characters in which crop we seen that particular plants only we have selected by. Already you seen in the plant breeding how we are going to select the parents among the different crop and their varieties which they are present in our locality. Understood? Or in our crop field we have to select some parents or select some plants as parental plants. Understood? Here we are not discussing anything about their sex, we are not discussing about their gender. Understood? So it is purely we seen only the plants what we have as chosen as parents for taking plant cells only. So we have taken protoplast from we have taken protoplast from parent A and protoplast from parent B. Now you please draw the diagrams of protoplasts. Okay, these are the protoplasts we are collected from plant cells. So simply the remaining cytoplasmic constituents are there here. Cytoplasmic constituents are there, but we focus the concentration on that structures which they are present in the plant cells, like vacuole, which is the larger sized structure which is present in the plant cell. Understood? Vacuole, and second one is nucleus. The remaining everything, this is the liquid portion and some other cell organelles which they are present, it is completely called as cytoplasm. You know what it is? It is the outermost layer of the protoplast. Outermost layer of the protoplast are naked plant cell or naked protoplast. It is called plasma membrane. In the same way, we have to collect one more protoplast from another parent B. Now got the point? Here also same, everything is as it is present, but genetically some differences are there. Genetically some variations are there. That's why those different variations now we are trying to incorporate into the single cell, what we are producing as a resultant cell through the process of somatic hybridization or protoplast fusion. So see here, as it is a vacuole, this is a nucleus, this is the cytoplasm, this is the outermost layer in the protoplast called plasma membrane. But what is the outermost layer of the plant cell? Plasma membrane. But what is the outermost layer of the plant cell? In the plant cell, outermost layer is cell wall. Already we discussed. Now got the point? Here we seen what cell wall has outermost layer, cell wall is as a outermost layer we seen here, that cell wall is going to be removed, cell wall removed, cell wall removed, that particular cells only we are calling what? Protoplast. What it is? Protoplast. Like that only we are collecting from other plant cell also, from other parent. It is the normal plant cell of parent B. Okay, it is a normal plant cell of parent B. Here also cell wall removed. Cell wall removed. That removed cell wall of plant cells we are taken as protoplasts. So it is the protoplast we have taken from parent cell or parent plant cell B. It is the parent plant cell B. It is the parent plant cell A. So just we have taken two individual protoplasts from parent cell A, from parent cell B. These two are the protoplasts now. These two are the protoplasts we trying to use in the process of somatic hybridization or protoplast fusion by using two techniques, either electro fusion, electro fusion or chemo fusion. Electro fusion or chemo fusion. If supposed to take it is occurs by chemo fusion. Just simply we are using, if it is occurs through the chemo fusion, we are using PEG or sodium nitrate. Sodium nitrate are the very special superficial chemicals which they are going to change the vicinity of environment, vicinity of the protoplasts, and these two are encouraged by the chemicals like PEG and sodium nitrate get fusion. These two are encouraged to fuse by motivating chemicals like sodium nitrate or polyethylene glycol during the process of chemo fusion. Then these two are slightly fused initially. First of all they fuses its cytoplasm. One cell to another cell they move very close, and when they make contact in between these two, parent cell A and parent cell B, these two are, especially we focus on nuclei of two cells. These two are going to be dissolved its membrane. What it is? Plasma membrane. This is the plasma membrane. These two are fused to dissolves. The dissolving ability taken by the chemicals what they are motivating, or electric field or charge what we are provided to the two different parent protoplasts. Then the membrane is going to be dissolves. Then these two are going to slowly, the cytoplasm of parent cell A and cytoplasm of parent cell B, these two are getting mixed. So this is the process we are calling what cytoplasmic fusion. Cytoplasmic fusion followed by nuclear fusion. So here these two are the cytoplasm. Yes, all these are the liquid material which we seen in the cells. Understood? The cytoplasm completely fused. Now these two nuclei ready to get fusion. That is under the process of nuclear fusion. So when the nuclear fusion is going to be taking place, then it is completely developing into a single hybrid cell. That nucleus A and nucleus B, these two are getting fused to make a single nucleus. These two are getting, means here both the genetic information is present, that is A plus B. Okay, the genetic material which is present, which is genetic sequence what it is present in the parent cell A or protoplast A, that genetic material which we seen in the protoplast B is going to be get fused to make a single nucleus. These two are going to be fused to make a single nucleus or single nucleus. single nucleus. This is the cell. It is a single cell with more diversified cytoplasm and diversified or diverse genetic material with the parental cells. It is a new cell is going to be produced. It is a new cell is going to be produced. That new cell is called hybrid cell. Hybrid cell. This hybrid cell developed from the somatic cells, that's what is called somatic hybrid or somatic hybrids. More number of cells we are producing now, thousands of hybrids we are producing. Okay, those are called somatic hybrids. These somatic hybrids okay, produced in the process of somatic hybridization. Somatic hybridization. Somatic hybridization. So these somatic hybrids, somatic hybrids first produced or obtained by a great scientist like Carlson et al. Carlson et al. In the year 1972. In the year 1972, a great scientist like Carlson et al. Who first prepared or made a new hybrid cell by this process, Protoplast Fusion or somatic hybridization. Okay, he done that somatic hybridization in between, okay, two different species of plants of same genera. That is, he made in the tobacco plant. So tobacco hybrid he made. In between two different species like Nicotiana. Note down, students. Nicotiana glauca versus Nicotiana Langsdorffi. Nicotiana Langsdorffi. In between these two only, he produced a new hybrid variety, okay, which is going to give more yielding. So like that, some scientists also they made another variety of okay, somatic hybrid plant only, that is called pomato. You please remember pomato. Here two different genera they are using. So this pomato is a hybrid species of two different genera. Hybrid species of two different genera. So this particular hybridization is intergeneric hybridization. Intergeneric somatic hybridization. You please remember. Intergeneric somatic hybridization is the method what they follow to produce pomato. Pomato produced by Protoplast Fusion done in between potato and tomato. So potato and tomato both are belongs to same family but two different genera. Okay, parent A, parent B. They have collected two different protoplasts, genetically diverse protoplasts from two different genera, and okay, they do somatic hybridization to produce tomato as a intergeneric intergeneric somatic hybridization they follow to give pomato, but this pomato, that somatic hybrid plant like pomato produced successfully, but it is not maintain desirable characters. Unfortunately, that pomato unfortunately did not have, note down students, unfortunately this pomato did not have desirable characters. So students, scientists made a new hybrid plant like pomato, a new hybrid plant like pomato. Scientists have been successful in the production of plants like pomato, but that pomato is not is not expressing desirable traits or characters. In that only they failed. That's why this pomato is not commercially valuable. This pomato is not we are utilizing as producing commercial products, or utilization of commercial products are not may not taken by the pomato. Pomato plant production is okay, but it is not producing expressing desirable characters in the form of products what we are expecting. But that expectations are not met by the pomato. It is unfortunately did not have the desirable characters or traits. So like that, dear students, finally what we understand, all these other techniques are very much useful for increasing plant number. Automatically the plant number with the desirable characters we increases, then automatically food production increases. If the food production increases, then automatically the scarce of food okay generally reduces, and hunger reduces, malnutrition reduces. From this topic finally we understood about the production of plants with the desirable characters and high yielding varieties of plants how we are producing, or otherwise by using somatic parts also, by following methods of tissue culture and somatic hybridization, how can we increase the plant number and plant productivity, and how we are going to producing that particular productivity of plants. Understood students? So all these are the different techniques and methods we follow to enhance the food production. This is the topic what we discussed. So applications of tissue culture you know very well. Applications of tissue culture is, the tissue culture is very much familiar for the increasing okay rapid clonal population. Rapid clones we are producing with the desirable characters in less time and less space. This is very important point, you please note down. Okay, tissue culture is very important application it is given, that is we produced rapid clonal population, or rapid clones they are producing within less time and less space. So that's what it is called micro propagation. Tissue culture is also called as micro propagation, and also it gives another method, that is meristem culture, through which we are producing a large number of clones without diseases, or disease free clones, pathogen free clones we are producing by method. It is called meristem culture, which is also one of the methods of tissue culture. So like next one is, it is very important, that is somatic hybridization, how we are going to get produced different hybrid varieties somatically. Understood? By using somatic parts. That's why it is called somatic hybridization. So with this topic we concluded, we finished our chapter strategies for enhancement in food production. Thank you students. Next we are going to discuss about our new topic, that is about microbiology. In the second year, it is one of the important topics for intermediate second year botany. So now we go to discuss about microbiology as our next topic. In the next lecture we are discussing about that chapter microbiology. Thank you.